Hi, my name is Christoph Becker. I am Professor of Molecular Gastroenterology at the Department of Psen1, University Hospital Erlangen in Germany. I'm also the spokesperson of the DFG-funded research center Transregio 241, where we focus on understanding the pathophysiology of inflammatory bowel diseases like Crohn's disease and ulcerative colitis. The main interest of our consortium is to gain a deeper understanding of the molecular and cellular processes that drive IBD. My own research group focuses on intestinal epithelial cells and their function not only as a barrier, but also as a mediator between the microbiome and the immune system.
Hi, my name is Lena Erkert and I'm a postdoctoral researcher in the group of Christoph Becker. In our recent publication in Gut, we made the surprising discovery that proteins previously associated with neurodegenerative diseases, such as for example Alzheimer's disease, play a crucial role in maintaining intestinal homeostasis.
To protect the digestive tract, intestinal epithelial cells create a strong barrier that blocks harmful microbes and other substances from invading the gut. Perturbations of epithelial homeostasis can lead to intestinal pathologies like inflammatory bowel disease. As of now, we don't fully understand the underlying causes of IBD, and the treatments available aren't curative. Instead, they focus more on managing the symptoms of the disease. That's why there is an urgent clinical need for more research to uncover the molecular mechanisms behind IBD so that we can develop new treatment options.
To uncover underlying mechanisms of IBD pathogenesis, we sought to identify novel and previously unrecognized pathways that might be involved in intestinal inflammation. Surprisingly, in mouse models of colitis, we identified an enrichment of pathways attributed to neurodegeneration, where we identified the Alzheimer's disease-related molecules, presenilins, as possible interesting protein candidates. While mutations in the presenilin genes are well known to be associated with familial Alzheimer's disease, little is known about their function in the gut. By analyzing the expression of Psen1 in different cohorts of IBD patients, we were able to show that Psen1 is significantly downregulated at both mRNA and protein levels in IBD patients, and that its expression levels correlate with disease severity. Interestingly, by using an integrated analysis approach, we observed that this downregulation occurred specifically in intestinal epithelial cells without affecting immune or stromal cells, emphasizing the role of presenilins in maintaining gut health during inflammation. Using inducible Psen1, Psen2 double deficient mice and advanced macroscopic and microscopic techniques, we were able to dissect the functional role of presenilins in intestinal homeostasis. Our data demonstrate that the presenilins are key factors for intestinal homeostasis, as dual deletion of Psen1 and Psen2 leads to the development of spontaneous intestinal inflammation in vivo, as seen by drastic body weight loss, signs of colitis in high-resolution mini-endoscopy, and various different microscopic analyses. Using organoid models as unique epithelial cultures, we further investigated how presenilins contribute to the maintenance of the intestinal barrier at a cellular level. Overall, our analysis clearly demonstrated a key role of presenilins in barrier integrity, where deletion of both presenilins lead to the breakdown of the intestinal barrier and bacterial translocation to distant organs, eliciting a systemic inflammatory response. To investigate whether barrier breakdown with subsequent bacterial translocation could be chiefly responsible for the severe wasting disease of mice lacking Psen1 and Psen2 in the epithelium, we pre-treated mice with an antibiotic cocktail to deplete the microbiota. Overall, antibiotic treatment of inducible Psen double knockout mice could improve signs of inflammation and barrier breakdown, but could not rescue the weight loss in these mice, suggesting that microbial translocation in these mice is a driver of colitis development. But there might be other potential mechanisms underlying the rapid and severe weight loss.
In addition to abdominal pain and diarrhea, 70 to 80% of hospitalized IBD patients and 20 to 40% of outpatients experience malnutrition. The factors that contribute to these observed effects in IBD patients are also poorly understood.
Using our genetically engineered mice, we were able to show that epithelial presenilins are essential for a balanced intestinal epithelial cell differentiation. More specifically, we could show that a loss of both proteins leads to an almost complete loss of absorptive enterocytes, a cell type that is responsible for the uptake of nutrients from our diet, such as glucose or lipids. This disruption ultimately leads to malnutrition in these mice. Molecular analyses of the underlying signaling pathways leading to all these observations revealed that Notch signaling, which is one of the most important signaling pathways in the gut to maintain homeostasis, was critically impaired upon deletion of presenilins. Overall, our study identified the presenilins Psen1 and Psen2 as previously unknown players in intestinal homeostasis and inflammation, where they act as key molecules in maintaining a balanced IEC differentiation and barrier function integrity to prevent malnutrition and intestinal inflammation. Due to these findings, we believe that supporting the presenilin enzymatic activity in the gut epithelium or downstream signaling may provide new targets for novel IBD therapies.